let's get started. It's my great pleasure today to introduce Dr. Aaron Gitler as our CTSI distinguished speaker for today. Dr. Gitler obtained his bachelor's degree from Penn State University and his PhD at UPenn, and completed his postdoctoral training at the Whitehead Institute for Biomedical Research in Cambridge. He then started his own lab, first at UPenn, and a few years later was recruited to Stanford, where he is right now. And he is a professor of genetics at Stanford and also an investigator at the Chan Zuckerberg Biohub. And Aaron's lab took a really unusual and innovative approach to use yeast to study neurodegeneration, in particular ALS. And you may ask, why yeast? Yeast doesn't even have a brain. Well, over the years, Aaron has published such a stellar body of work that have convinced the skeptics that if you look at the level of protein function and protein-protein interactions, yeast and mammalian cells actually have a lot conserved and in common. And using this approach, Aaron has made many important findings for the mechanism of action of genes involved in ALS, and also identified new genes that's contributing to ALS. And his research started with yeast, but doesn't stop there. And he actually carried out really detailed mechanistic work in mouse, and also using the power of human genetics to further lead to more clinically relevant study. And one of the really important findings in the ALS field in the past few years is Aaron's discovery of ataxin-2 as a ALS risk gene. And his lab has found that medium length expansion of poly-Q in ataxin-2 is one of the most common ALS risk factors. And let's see, if I look at my little cheat sheet. So with this ataxin finding, he actually provided human genetics validations and found that and this work has led to the initiation of a clinical trial in humans to test ASOs targeting ataxin-2. And Aaron's lab has been leading innovations on multiple fronts, and he has published 160 papers. And obviously, I'm not going to go through those discoveries. And he has won so many awards. There's a long list, like a whole page of awards in his CV, which I'm also not going to go through. But just to highlight a few, he won the top 5 dissertations nationally award for his PhD work, and the NIH Director's New Innovators Award, and the NIH R35 Research Program Award. In addition to being an outstanding scientist, Aaron also is great at building communities and helping people in the neuroscience community. And he's been very generous with his time and offering career advice to junior researchers and connecting them to opportunities and highlighting their work in the many fantastic meetings and seminar series that he has organized. And I'm among many grateful people who have received help from Aaron. And Aaron has also organized some high quality meetings, both online and in person. And during the pandemic, he has organized a Neuro Zoom series, and I'm sure many in the audience have attended. And that was like something that I was looking forward to every week during the pandemic. It has so much inspiring science, and it helps us go through the challenges of the pandemic. Well, without further ado, let's join me in welcoming Dr. Gitler.
Thanks a lot, for inviting me and for the introduction. It's been great to get to know you first at Stanford and now your research here. Thanks for inviting me to visit, and happy to share with you some of the things we're working on in the lab and some of our new directions. So my laboratory is studying the mechanisms of human neurodegenerative diseases. And these diseases are increasing in prevalence as our population ages. These diseases have very different clinical presentations. Some of them affect memory, like Alzheimer's disease, whereas other ones are a movement disorder, like Parkinson's disease, and cognitive ability can be relatively preserved. So different phenotypes in different diseases. But despite those differences, there's a common theme that unites all of these neurodegenerative diseases, and that's protein misfolding. So proteins in the brain of patients with these diseases become misfolded and start accumulating and aggregating. And sometimes they can accumulate outside of neurons, like in amyloid plaques in Alzheimer's disease. Sometimes they can accumulate within neurons, like neurofibrillary tangles in frontotemporal dementia. They can even accumulate in the cytoplasm. Those are alpha-synuclein aggregates called Lewy bodies in Parkinson's disease. They can even be right in the nucleus. This is a polyglutamine aggregate. That's the hallmark of Huntington's disease. So the idea is that different proteins can aggregate in different parts of the brain. And the mission of my laboratory is to define the cellular pathways that are affected when these proteins misfold. I also think it's an interesting basic cell biology question because these proteins and the genes that are encoding them are expressed throughout the brain. Some are even ubiquitous, yet the diseases are associated with a very selective vulnerability. Some neurons are lost and others aren't. So we hypothesize that these proteins, when they misfold, interfere with certain cellular pathways that some neurons are more susceptible to than others. And if it's true that these diseases are deeply rooted in basic cell biology, we think we could use simple model systems to study them. And as introduced, the model that we started with is the budding yeast. It's a very simple model system, but I'll show you that there are many powerful experimental advantages that we can learn the key cellular pathways that are affected by each of these disease proteins. Perhaps the main advantage is it's very fast, where these diseases, if you study them in people, will take many decades, and you can study them in mice, they'll take a couple years. But I'll show you experiments where I can take one of these human neurodegenerative disease proteins and introduce it into yeast. In just a couple hours, it'll start aggregating and then eliciting phenotypes. And then we can use those phenotypes to perform modifier screens to look for genes that could suppress or enhance the phenotype. And then we validate some of our findings in other model systems. And recently, we've been extending studies to mouse models and analysis of human patient samples. I'll share that with you. So today, just going to talk about one neurodegenerative disease. It's called amyotrophic lateral sclerosis, or ALS. Even if you're not interested in this disease, hopefully you can see the approaches we're taking and the questions we're asking, because I think this approach could be applicable to other disease situations. So ALS is a disease of motor neurons. It's motor neurons in the brain and the spinal cord. When the motor neurons from the spinal cord innervate muscle and control all movement. When these motor neurons die, it causes muscle weakness, and then that unfortunately progresses to paralysis and then ultimately death very rapidly. The disease is a fast progressing disease, 3 to 5 years after onset. It's a universally fatal disease. And like other neurodegenerative diseases, ALS also has misfolded proteins. So this is a section through a spinal cord of a patient who died of ALS, and you can see protein aggregates accumulating in the motor neuron. About 2% of all ALS cases are caused by mutations in a gene called SOD1. If you have SOD1 mutations, this protein that aggregates here is a mutant form of SOD1. But those 98% of cases that don't have that SOD1 mutation, they also have protein aggregates, but it's not SOD1, it's a different protein. And this protein was discovered in 2006 by my colleagues at University of Pennsylvania, Drs. John Trojanowski and Virginia Lee, to be an RNA binding protein. It's called TDP43. It's a nuclear protein. It regulates splicing, other aspects of RNA metabolism. Somehow it gets depleted from the nucleus and it starts accumulating out in the cytoplasm in these round Lewy body-like or these skein-like aggregates. This is the main pathology seen in all ALS cases except those that have that SOD1 mutations. Very shortly after they discovered TDP43 pathology, geneticists identified mutations in the gene that encodes TDP43 as a cause of rare forms of ALS. So it's this conjunction of pathology and genetics that's now focused attention on TDP43, trying to figure out how it's contributing to disease. This slide shows the questions I want to talk to you about today. These are 2 sections of human spinal cord. I stained them with an antibody against TDP43. You can see on the left the normal situation. You can see the nucleus, you can see the nucleolus. You can see TDP43 is sort of diffusely localized throughout the nucleus. So you can imagine it's binding to RNA, regulating alternative splicing. This is its normal function. But then on the right is the disease situation. Now you can see a few things going on. You could see these large aggregates of TDP43 building up, or these filamentous ones. You can see smaller brown dots, these smaller inclusions. Maybe disease is caused by TDP43 causing some sort of toxic, like gain of toxicity where it aggregates, it interferes with RNA binding proteins. But then if you look at the nucleus, you can see that now there's no more TDP43 in the nucleus. It's depleted from the nucleus. So maybe disease is actually caused by a loss of TDP43's function. Those two hypotheses are not mutually exclusive. Both could be going on, but we just have to think about those as we're thinking about mechanisms. So the first experiment that I did in my laboratory was to see if I could use this yeast system to learn about some of the mechanisms by which TDP43 might aggregate and become toxic. So we took human wild-type TDP43 and expressed it at very high levels in yeast cells under the control of a very strong but tightly regulated promoter, the galactose-inducible promoter. So we can grow our yeast cells in glucose. It represses expression of TDP43, and then switch them to galactose, and it turns on expression. So you can see here, this initial set of experiments, TDP43 formed these cytoplasmic aggregates when we fused it to GFP, and then when we performed a spotting assay where we take the yeast cells, serially dilute them, and then spot them onto agar plates, you can see that they can form a colony if they express GFP, but it was toxic when they expressed TDP43. And then on the left is just a negative control where they're not expressing anything, just to show you that I spotted the same numbers of cells. So initial experiments in the lab recapitulated two important features of TDP43 that I think are relevant for disease: the cytoplasmic aggregation and then this toxicity. And then I use this model over and over again to ask stuff like, what parts of TDP43 are sufficient and necessary for aggregation and toxicity? What are those mutations that cause ALS due to it and stuff like that? But the main reason I wanted to use this yeast model was to figure out the mechanism of toxicity. I wanted to figure out why is TDP43 killing yeast cells? I didn't care why it was killing yeast cells in and of itself, but I thought if I could figure out why it was toxic in yeast, that would give me like a toehold to ask how it could be toxic in neurons. And this is really why you use model systems. So yeast cells have about 6,000 genes. We have in our laboratory a library of plasmids where we can overexpress all of them one by one. About 4,850 of these genes are non-essential. We have single knockouts of each of those. Even the essential genes we now have conditional alleles for. So you can ask what genes, when overexpressed or knocked out, can modify the phenotype. The logic is like if TDP43 is toxic, say, by interfering with like some essential enzyme in a cell, you can imagine if you overproduce that enzyme, you could suppress the toxicity. You can imagine if you overproduce another inhibitor of that enzyme, you might make it worse. So if you do the right negative and positive controls, enhancers and suppressors of toxicity are both informative. This is an example of like one of the results from the screen. We have a large agar plate. Each spot has yeast cells expressing TDP43 and then overexpressing one gene from the yeast genome. We do all 6,000 genes in triplicate. Most had no effect, but you can see like this one up here allow the cells to grow better even in the face of the toxic TDP43. So this is a suppressor of toxicity. These two ones made it grow worse. One worry I had about this type of screen is I'm putting a foreign human aggregation-prone protein in yeast. I'm going to get all these unspecific effects. And when I was a postdoctoral fellow at the Whitehead Institute with Dr. Susan Lindquist, I did the same screen, but for this other protein called alpha-synuclein. This is a protein that aggregates in Parkinson's disease. And I was just worried, like now in my own laboratory, I would just get all the same things that I got as a postdoc. But what was cool was that, like, even though they both look the same when you express them in yeast, alpha-synuclein and TDP43, same library, same screen, same robot, me doing both screens myself, I get almost non-overlapping hits. So I think, so I'm excited about the specificity because it means that we're not getting non-specific things. It means that, I think it means that the hits we're getting are related to the underlying biology of these disease proteins. So we have about 40 genes that when upregulated can suppress or enhance TDP43 toxicity. And whenever you do one of these screens, like a yeast screen or Drosophila or RNA-seq or single-cell RNA-seq or mass, whatever, you always get a list of hits. And it's always a challenge to figure out like which ones on that list to work on. So I'll just tell you a story about one gene that we found in the screen and just how we've gone in different directions with it. It's a gene called PBP1. And we found that when we overexpress PBP1, it enhanced TDP43 toxicity. Yeast cells express TDP43 grow slower. If they also overexpress PBP1, they grow even slower. This did not worsen the toxicity of alpha-synuclein or a mutant fragment of Huntington. Conversely, if I delete PBP1, this was now sufficient to suppress the toxicity of TDP43. Wild-type cells grow slow when they express TDP43. If you delete PBP1, this suppresses the toxicity. We focused on PBP1 because it had a human homolog that had already been implicated in another neurodegenerative disease called spinocerebellar ataxia 2. The gene is called ataxin-2. It's a member of a family of polyglutamine disease genes like Huntington. And luckily for us, my colleague at the University of Pennsylvania, Dr. Nancy Bonini, had been studying ataxin-2 in Drosophila for many years. And with her, we've been making flies to study TDP43. So we just said, hey, let's just cross these two flies together. So in this experiment, we direct transgenes to the fly eye. This is the fly eye, the section to the fly eye. Very mild degenerative phenotype if you express TDP43 at moderate levels. No effect if you upregulate ataxin-2. But when we combine the two, it causes this massively degenerating eye. And then when we lowered the amount of ataxin-2, that was sufficient to rescue this mild phenotype. You can then direct the transgenes throughout the nervous system, and this caused flies to live shorter if they express TDP43. If they expressed ataxin-2 on its own, no effect. Together with TDP43, they lived shorter, and then we could lengthen the survival of TDP43 expressing flies by lowering the amount of ataxin-2. This was cool because this meant that that genetic interaction that we found in yeast works like the same way in the nervous system of an animal. And remember I said it was one of these polyglutamine disease genes like Huntington. So what that means is ataxin-2 has this trinucleotide repeat, CAG, which encodes glutamine, abbreviated Q or polyQ. And there's always, for ataxin-2, 22 of these. If it somehow gets expanded to greater than 34, it causes spinocerebellar ataxia 2. So 22, rarely 23 is the normal length. Greater than 34 causes SCA2. But because I found it in a screen with an ALS protein, I was thinking, could this gene be connected with ALS? And I was thinking, what about something in between, like a length of Qs longer than normal, but not like over the range to give you SCA2? So to test that hypothesis, I collected about 1,000 ALS patient genomic DNA samples, about 1,000 controls. No one in my lab wanted to do this, so I did this experiment myself. I made primers flanking this region, and this is like a gel picture from my lab notebook. I just cut the bands out of the gel and TA clone them, and then send them for sequencing, and just can count the CAGs. Eventually, one of my colleagues told me that I could send the amplicons, if I incorporate a fluorescent dye during the PCR, I can send the amplicons for sequencing by fragment analysis by capillary electrophoresis. And then here, that works much better. So you can see in this individual, both alleles of ataxin-2 have 22 CAGs, so there's one peak. This one is 22, one is 23, so there's two peaks. The ALS cases, I started to see expansions at like 27, 30, 32, 33. And then this table just summarizes the data. So 980 healthy individuals, I only found repeats greater than or equal to 27 in 14 individuals, but I found almost 5% of ALS cases having these intermediate length ataxin-2 expansions. So we propose that these intermediate polyQ expansions and ataxin-2 increase risk for ALS. What's been exciting is the last few years, laboratories around the world have been replicating these findings in independent ALS populations, like in Europe and Asia, replicated in North America. So it appears to be a real powerful genetic contributor to ALS. It's also, I think, cool because it comes from a simple model system. So I think it underscores that model systems can be powerful to learn about biological processes that may be important for disease. So now I'm obsessed with ataxin-2 because we want to figure out what its normal function is. We want to figure out because like if it has 22 glutamines, it's the normal length, it's the normal function. But if it has like say 40 glutamines, it causes Purkinje neurons in the cerebellum to degenerate. But if it has 30 glutamines, it causes motor neurons to degenerate. So I really want to figure out what at the biophysical, cell biological level, what is the difference between those three ataxin-2 proteins? And from some of our studies of individuals who have those intermediate expansions from their cell lines, we know that ataxin-2 harboring those intermediate polyQ expansions is more stable. So more stable ataxin-2 is associated with increased risk for the disease. And then the stuff in yeast and flies lowering ataxin-2 protected against TDP43 that suggested that maybe we want to target ataxin-2 as like a therapeutic target. So to test that, you need to do experiments in mouse. So these are experiments that Lindsay Becker, a graduate student in my lab at Stanford, performed. She's now a postdoctoral fellow with Cliff Brangwyn at Princeton University. So what Lindsay did was she crossed a transgenic mouse model that expresses TDP-43 throughout the nervous system under the Tsai-1 promoter control. This is a very aggressive model of degeneration. These mice become paralyzed and die by like 30 days at very high levels of TDP-43 expression. And then she crossed these to 2 independent lines of ataxin-2 knockout mice generated by independent targeting strategies. Those mice are viable, fertile, and she analyzed wild-type heterozygous and homozygous knockouts. So the TDP-43 transgenic mice should die by day 30. And she found that mice that were heterozygous for the ataxin-2 knockout had a significant extension in survival. But then when she looked at the homozygous knockouts, they had this really dramatic extension with some mice living over 300 days. Actually, some live still over 600 days, whereas all the litter mates died by day 30. This was also sufficient to affect the pathology in the spinal cord of these animals. These are sections of the spinal cord. These are phosphorylated TDP-43 accumulations in the wild-type mice that are reduced and eliminated in the heterozygous and homozygous ataxin-2 knockouts. This is the quantification. We really can't find any TDP-43 pathology in the spinal cord of the ataxin-2 knockout mice. So we next wanted to see if we could test something that might be more clinically applicable. So we collaborated with a pharmaceutical company, Ionis, to test antisense oligonucleotides targeting ataxin-2. These are molecules, DNA-like molecules that bind to an RNA you design them against. And all of our cells have an enzyme called RNase H, which recognizes this DNA-RNA hybrid and then catalyzes its destruction. We delivered these ASOs, or one targeting ataxin-2 or one as a negative control scrambled sequence, into the nervous system of these animals at day 1. And we found that a single administration at day 1 was sufficient to lower ataxin-2 RNA and protein, and also have a significant extension in survival in the ones injected with the ataxin-2 targeting ASO. This video is anecdotal, but it shows you that the rescue can be strong. These are 3 of the TDP-43 transgenic mice at day 20. We injected all 3 of them at day 1. 2 of them we injected with like the negative control scrambled sequence. Those mice are paralyzed. We have to euthanize them at this point. This one we got. This one is unimpaired, goes on and lives much longer. This one we injected with the ataxin-2 targeting ASO. So we're excited about the power of this ASO approach to rescue these animals, but we're really excited that, I said this before, but I'll show it as a slide. TDP-43 pathology really is the major pathology in virtually all ALS cases, except those rare ones that have SOD1 mutations and then this other RNA binding protein. So we hope that targeting a modifier gene could be broadly effective in diseases involving TDP-43. We're really inspired by the use of antisense oligonucleotides as a therapy. This last year, there was approved by the FDA for targeting a mutant of SOD1 in familial ALS. There was some trouble with one targeting the C9orf72 mutation. Actually, a couple days ago, so Biogen's been testing this ataxin-2 ASO in humans. Just a couple days ago, they reported that they're able to lower ataxin-2 levels, but after 6 months, they don't see any clinical improvement. We could talk about what that means, and we're still trying to digest that. There's also one that's targeting mutant fragment of FUS in a very aggressive juvenile form of ALS that's not published yet, but it's looking really really promising in those rare but aggressive forms of the disease. So we hope that these ASOs work, and we hope that strategies to lower ataxin-2 works, but we think we need to learn more about ataxin-2 regulation, because maybe we can come up with other ways of lowering its levels. So I want to tell you about 2 screens that a graduate student and a postdoctoral fellow in my laboratory have been performing, trying to learn how ataxin-2 is regulated to come up with other ways that we might want to lower its levels. So first, Caitlin. So Caitlin, she engineered a human cell line with CRISPR to have this high-bit tag in frame with ataxin-2. High-bit is an 11 amino acid fragment of nanoluciferase, and it's inert. When you add large bit, it reconstitutes the luciferase and emits light, and then it works as a cellular readout of ataxin-2 levels, because if she knocks down ataxin-2 with siRNA, this is sufficient to lower the luciferase signal. So she miniaturized this and did a genome-wide siRNA screen, knocking down all human genes, looking for genes that would knock down have less ataxin-2 expression. And after filtering everything, the top hit she found was RTN4R, which is also known as the no-go receptor, which has been implicated in axon regeneration. You can see here, if she knocks down ataxin-2 with siRNA, it lowers ataxin-2 levels, of course, but when she knocks down RTN4R, it lowers ataxin-2 levels just as much. And this is the immunoblotting. So she figured out the mechanism. So it appears that RTN4R knockdown works at the level of the protein because it doesn't affect mRNA levels. And when she blocks the proteasome with MG132, this interferes with the ability of RTN4R knockdown to lower ataxin-2 levels. There's a peptide antagonist of the no-go receptor called NEP1-40. When Caitlin adds this to cells, it very rapidly lowers levels of ataxin-2. She then tested this in neurons, either from mouse or from human iPS cells. And in both cases, knocking down RTN4R can lower ataxin-2 levels in mouse and human. Same thing with this peptide inhibitor. It lowers ataxin-2 levels. And then finally, when she analyzed RTN4R knockout mice, she sees a reduction in ataxin-2 in the brain of the heterozygous and the homozygous knockouts. So perhaps targeting RTN4R could be another way of interfering with ataxin-2 expression. The next screen is from Garam. Same idea, looking for regulators of ataxin-2. So Garam is a pretty amazing graduate student in my lab at Stanford. She just left to be a postdoctoral fellow at the Broad Institute. But before she was a grad student at Stanford, she was a professional concert solo violinist. And but not just like a solo violinist, like a world-renowned solo violinist. And I'm saying that because like to be at that level, what it takes in terms of like dedication and practice, hard work, off the charts with her. And even in basketball, she plays basketball like 2-3 hours every day, and she's the best in the lab at basketball. So she sort of brings all of that. That was like a condition of joining the lab, if she can play basketball with the guys in the lab. And like she brings all that to the screen that I wanted to tell you about. So she wanted to look for regulators of ataxin-2 levels, but she didn't want to use tags or anything. She wanted to do a FACS, fluorescence activated cell sorting based screen to look for levels of endogenous ataxin-2. I didn't think this would work. Ataxin-2 is not a cell surface protein, but she made an exhaustive allelic series of cell lines that have all different levels of ataxin-2 expression, got an array of antibodies, and calibrated the system to detect different levels of ataxin-2. And then she used a genome-wide CRISPR library and it transduced a human cell line that expresses Cas9. She has 10 guide RNAs per gene, 20,000 genes, plus 10,000 negative control guide RNAs, all barcoded, all pooled together. She stains the cells with ataxin-2 antibody and then sorts them for ones that have high or low levels of ataxin-2, and then isolates the DNA to figure out which guide RNAs did what. You can see some of her validations. She finds hits that when knocked out, lower ataxin-2 as much as the ataxin-2 knockout. And then the hits that she was most excited about is that she got every subunit of this lysosomal vacuolar ATPase or VATPase. Whenever you do a genetic screen and you get every subunit of a complex, it tells you that there's something probably important about that complex. Individual subunit knockouts lower ataxin-2 levels. This seems specific to ataxin-2 because when Caitlin knocks out the subunits, it doesn't affect other poly-Q proteins like ataxin-1 or Huntington. It lowers ataxin-2 levels specifically. She also became interested in this target because it's already a target of a known small molecule that's FDA approved for the treatment of a bone disease and also for osteoporosis. It's been approved over 40 years, so there's decades of like real world safety data on it. So she got this compound in, and she found of course it's very safe, and she adds it to neurons, and it across a wide range of concentrations lowers levels of ataxin-2. Same with human iPS-derived neurons. And then in ongoing experiments, she's been feeding this to mice in their water and then testing for lowering levels of ataxin-2 in the brain. And we're trying to like modify this compound to see if we can improve its properties. But we're very excited about these types of screens. Just look, all of our previous screens have been looking to work on all the pathways, but maybe at the end of the day, we just need to lower or raise a disease gene a little bit, and figure out the regulators of it. So Olivia Gauthier, a graduate student in the lab, became interested in an ALS gene called TBK1. This is a haploinsufficient, so we might want to increase its levels. So she did a genome-wide CRISPR screen using Garam's approach and found a knockout of a phosphatase that increases levels of TBK1. And then now pretty much all of the postdocs in the lab are doing their own CRISPR screens, and so more to come on that. Okay, so back to this thing that I started with loss of function and gain of function. I think a lot of the stuff with ataxin-2 probably involves TDP-43, ataxin-2 interactions out in the cytoplasm. But what's going on when you lose TDP-43 from the nucleus? So as soon as TDP-43 was discovered as the RNA binding protein, the pathological protein, it was an RNA binding protein, the first thing people wanted to do was figure out what RNAs does it bind to. So Don Cleveland and Jean Yo, together with Magda Polymenidou, Clotilde Gertran, they did a ClipSeq in mouse, just looking for all the targets that TDP-43 could bind to. And Yarne Ule's laboratory simultaneously did this in human brain and human neuronal cells, and they found thousands of RNAs that it can bind to. And then there was a very important paper from Phil Wong's laboratory in which he found an important function of TDP-43 is to repress cryptic exons. These are sequences that are in introns that should not be included in the mature messenger RNA. TDP-43 sits down on these yellow GU repeats and makes sure that these cryptic exons don't get included in the mature messenger RNA. When you lose TDP-43 from the nucleus, these cryptic exons creep into the messenger RNA, and they can cause problems for a few reasons. They could destabilize the RNA. They could introduce frame shifts. They could maybe make new peptides that aren't predicted. Importantly, these cryptic exons are not conserved. So if you do this experiment in mouse, you get a bunch of cryptic exons. If you do it in human, you get a bunch of cryptic exons, but they're different ones. The theme is conserved, the function is conserved, but the specific cryptic exons are not. Meaning that if you want to find maybe the disease-relevant ones, you need to do experiment with a human system. So Kevin Egan's laboratory and Don Cleveland's laboratory identified one human-specific one, a very important one called STAFMIN2. This is a abundant neuronal gene that the protein can bind microtubules, and unfortunately, in its first intron, there's a cryptic exon, exon 2A, that has a TDP-43 binding site right next to it, and a premature polyanylation signal, and a stop codon. TDP-43 sits down there, exon 2A never gets incorporated, but when you lose TDP-43, now this cryptic exon gets inserted and you make a truncated STAFMIN2. We were sort of wondering, like, could there be more of these cryptic exons out there, and could we find them? So this project started during COVID, when we were just at home. We just could read papers and analyze data. And the star of this project is Rosa Ma. She's a brilliant grad student in Stanford in genetics, literally brilliant in like the literal definition of the word. She read this paper from Eddie Lee's laboratory at the University of Pennsylvania, in which they had taken human ALS FTD patient brain samples, stained them with an antibody against TDP-43, and then isolated the nuclei, neuronal nuclei from those brains, and then sorted them into groups that had TDP-43 or did not have TDP-43, extracted RNA and did RNA sequencing, and found all the changes in gene expression associated with loss of TDP-43 from the nucleus. So Rosa wasn't interested in changes in gene expression. She thought she could take their data and reanalyze it, and now find changes in alternative splicing and look for maybe cryptic exons that were now present in the brain or the nuclei of patient brains lacking TDP-43. So she used two different splicing analysis pipelines, and she found 66 cryptic splicing events that occurred in the nuclei lacking TDP-43 in the brain of patients. She found STAFMIN2 again, and then she found this one, POLDIP3, which had been known as a target of TDP-43. But then she found 64 novel ones. And the one that immediately caught our attention is this one, UNC13A. This one caught our attention because This is the top GWAS hit for FTD and ALS. If you don't include the chromosome 9 locus, which we now know is the C9 ORF22 gene, UNC13A always comes out on top. This is in ALS. This is in FTD ALS. Again, this is C9 ORF22. This is UNC13A. And then this is the meta-analysis where UNC13A. So we've known about this for a long time, but no, we've never known like how SNPs somewhere in UNC13A can contribute to risk for disease. I think Rosa's figured it out. So this is the RNA-seq traces from those 7 samples from that paper from the web she downloaded. So this is exon 20 of UNC13A. This is exon 21. You see RNA-seq reads in the exons, of course, not in the introns. But when you lack TDP43, now you see RNA-seq reads in the introns. She calls these CE or cryptic exon. This suggested that TDP43 was directly repressing this cryptic exon. To test that, she performed experiments in neuronal cells using shRNA to knock down TDP43 and quantitative RT-PCR to detect the cryptic exon. She never sees the cryptic exon normally, but when you knock down TDP43, the cryptic exon goes up. The full-length transcript goes down. We think by a nonsense-mediated decay mechanism. And then by immunoblot, the protein is diminished. She also did this in human iPS-derived motor neurons, 3 independent lines, knock down TDP43, cryptic exon goes up, full-length transcript goes down, and the protein's eliminated. So UNC13A is a member of a family of proteins originally discovered in C. elegans because the mutants are uncoordinated because they have defects in neurotransmitter release. My colleague at Stanford, Dr. Tom Sudhoff, discovered the mouse version, MUNC13-1. Those knockout mice are born, but they die right after birth, and they are impaired in synaptic transmission. So this is an important neuronal protein, and it seems that now in neurons lacking TDP43, its expression is diminished. We next expanded our study beyond those 7 samples Rosa downloaded from the web. We collaborated with Dr. Len Petrocelli at the Mayo Clinic, and we analyzed every one of the samples in the Mayo Clinic brain bank that have TDP43 pathology. We can detect UNC13A cryptic exons in all of those. We looked in the New York Genome Center brain bank. Here, I think we found it again in FTLD, TDP, right brain regions. But I think where we don't find it is important. We don't find it in cases with tau pathology or SOD1 mutations or FUS pathology, never in the control. So I think it's not just these cryptic exons are appearing due to degeneration. It's specific to loss of TDP43. And so this RT-PCR analysis is good, but we wanted a way of visualizing the cryptic exons in the brain, in single neurons in the brain of a patient. So Rosa developed a single molecule fluorescence in situ hybridization approach for UNC13A cryptic exon. And she made a probe that flanks the canonical exon 20 and then the cryptic exon. This was key. And then she stained sections of brain with an antibody to TDP43, and then the red signal will be the cryptic exon. So you can see here, this nucleus has TDP43. This is normal, no cryptic exon. But these 1, 2, 3, 4 nuclei are in neurons harboring TDP43 pathology, and all of the 1, 2, 3, 4 harbor these cryptic exons. Every one we look at has the cryptic exon. The control neurons with normal TDP43 never have one. And then Rosa made a control probe. This is flanking the canonical exon 20, exon 21. Here, lots of UNC13A messenger RNA in a neuron with a normal TDP43, less of it when there's TDP43 pathology, consistent with her RT-PCR results. Remember I said it's a top GWAS hit for FTD ALS? So we're just looking at where the SNPs are, and the lead GWAS SNP is right in the cryptic exon. So, I mean, this gene is enormous. The SNPs could have been anywhere, but it's right in the cryptic exon. So we were very lucky. It suggested a hypothesis that maybe these SNPs increase risk for disease by making this cryptic exon more prone to be included when you lose TDP43. So Rosa's very clever. She realized that some of the samples she downloaded from the web could be from individuals that were heterozygous for the risk and the reference allele. And she looked for allele imbalance in her RNA-seq reads. So indeed, she found more RNA-seq reads harboring the cryptic exon in individuals harboring the risk allele. And then she did this systematically across the Mayo Clinic brain bank. And you can see as you go from 0 to 1 to 2 risk alleles, you get more and more UNC13A cryptic exons accumulating. And this is a survival curve she did. This is pretty crazy. So this is survival of humans with FTLD, just stratified based on their gene UNC13A risk haplotype. So if you have one risk allele, you live shorter, but even much shorter if you have two risk alleles. I don't want to overinterpret this result, but I think it means, because we now know how this cryptic exon, if we can block this single cryptic exon event, we should have at least this much survival benefit. I think more, because these individuals also harbor this risk allele. So a few takeaways. So can we define cryptic exon profiles that represent different ALS FTLD subtypes? So all of these cryptic splicing events are playing out across the brain. Maybe, but the diseases are very heterogeneous. Maybe different combinations of them can help us think about subtypes. Are there genetic variations in the genome that promote or protect against cryptic exon inclusion? UNC13A is the first one, but there could be other ones. We're looking for those. We think of these as like an Achilles heel. So having that SNP in UNC13A doesn't cause ALS, but maybe if there's some other trigger that causes TP43 dysfunction, now you're more prone to drive disease. This is sort of conceptually easy. Therapeutic strategies to block this. We know exactly where this cryptic splice acceptor site is. Can we just make a like a splice blocking ASO to do that? This is what I'm most excited about. I have no data at all on it. But what if some of those cryptic exons get turned into peptides, right? And like and then elicit some sort of T cell response. So some of these cryptic exons, most like the UNC13A1, SATHMA2 get degraded, but there are other ones that make in frame. And like every week at lab meeting, I'm always trying to like encourage people to work on it. I think this is an exciting avenue. And then there's other, like it turns out a lot of the targets we found are involved at the synapse, and a lot of the postdocs are working on those things. So that's going to be exciting. The last 2 minutes, I just want to tell you, like this will be completely lab meeting style. Haven't figured out how it works, all unpublished, but Yi Zeng, a postdoctoral fellow, he has a news story that I think is cool related to this. So he started this project like on his postdoc interview, because he started analyzing data with some other guys in the lab. And he's interested, and he was helped out with Anastasia, research specialist, and some postdocs, a technician, a high school student. So Yi became interested in polyadenylation. So polyadenylation is a level of gene regulation, and there could be alternative polyadenylation sites to produce same protein but different length 3' UTRs. This could affect RNA stability, localization. And then you can imagine if the polyadenylation site is internal, you might get a truncated RNA and protein. So there's some inklings that TDP43 can regulate its own 3' UTR, and there's some evidence if you look at genome, like patient data sets, that there might be alterations in this, but nothing really connecting TDP43 directly to it. So remember Rosa reanalyzed that data set from Eddie Lee? Well, Yi like reanalyzed it, but this time looking for alternative polyadenylation events. And he looked for in those. So I think this really means there are a lot of great data on the web that's just like out there to find and use. So he found there's data, and he found evidence that in neuronal nuclei lacking TDP43, there's lengthening or shortening of UTRs. He then went back to some of our in-house RNA-seq data sets with TDP43 depletion or other ones he found on the web, and he sees many examples of widespread alternative polyadenylation changes. But doing that with RNA-seq data is hard for a number of reasons. So he used a new technique called 3' N-seq, which gives him base pair resolution of exactly where the poly A site is. It lets you not rely on annotations of poly A sites. So you can find novel ones, and you can also find premature polyadenylation. So now he's just going to town using this, and you can see this is the RNA-seq data, broad peaks, but here you can see very precise peaks where the where the poly A site is. He can detect the STAFN2 polyadenylation event, premature polyadenylation event. That's you can't detect it with RNA-seq data. So he starts finding all manners of these like alternative blast exons, 3' UTR extensions, premature polyadenylation, coupling of
cryptic exon inclusion and premature polymethylation. So I just want to tell you, like, there are many interesting things here. I just want to tell you the one that I'm most interested in. Again, this is lab meeting style. There's a lot of different observations. We haven't worked out a coherent story yet. He found that knockdown of TDP-43 causes a lengthening of the 3' UTR in a gene called TMEM106B. So this is from the Eddie Lee data set. We could see longer 3' UTR, and then we teamed up again with Len Petrocelli's lab, looked at patient brains. Indeed, they have longer 3' UTRs when there's TDP-43 pathology. Why TMEM106B? So there was maybe 15 years ago a genome-wide association study done for FTLD with TDP-43 pathology, and there was only one hit, and it was TMEM106B. No one really knew its transmembrane protein, what its function was. And then Dr. Eisenberg and others in the last couple years had these stunning papers looking for amyloids in brains of patients with these diseases and finding it's a fragment of TMEM106B that forms amyloid fibrils and solving the structures of these in beautiful structures. And then in human genetics, it's very hard to go from a GWAS SNP and know what's the causative thing that's doing it. We were very, very lucky with Rosa's thing that the SNP was right there. It's never really that easy. It turns out that because of a phenomenon called linkage disequilibrium, where the SNPs can be connected to other SNPs you don't know about, we and other groups simultaneously found that there's a 3' UTR insertion linked to these SNPs. So this is right in the region that we find extended when we knocked on TDP-43. This, again, it's just an observation. Then we looked and we did a knockdown of TDP-43, and we find that TMEM106B dimers are reduced when we knocked on TDP-43. So we're trying to figure out, what does decreased dimers mean? And could somehow a reduction in dimers promote the amyloid formation? And how does this 3' UTR insertion that's a risk, how does that connect? We're trying to figure that out. But we became excited about the dimers because Len Petrocelli had this paper last month or 2 months ago, in which he looked at the correlation between the protective or the risk SNPs on forming this insoluble material. You could see individuals with this protective allele do not have insoluble material, and the ones that have the risk do have lots of it. I should mention, unlike other GWASs, for FTLD, if you have a granulin mutation, you get frontal temporal dementia. If you have this genotype, you're almost completely protected from it. So it's a very powerful protective effect. But I just looked down at this part of their Western blot that they didn't really focus on. I looked at the dimer. You can see the risk, less dimer, protective, more dimer. So now in our data, we knocked on TDP-43, we get a longer 3' UTR, less dimer. Here, if you're protected, you have more dimer. So something's good about having a dimer. We haven't figured out what it is. We did this one experiment. Again, I'll just show you the data. We don't know exactly what. Well, I'll show it to you. So we expressed TMEM106B open reading frame with no UTR, a short one, or a long one. The long one, we mutated the short site just to force it to be a long one. And we see reduction in dimer. Okay, that was good. But then we see this mystery band, a very short mystery band. We didn't really know what that was. And we knew from the structural studies that, so this is a transmembrane protein, TMEM106B. It's heavily glycosylated. So we thought maybe that mystery band could somehow be a unglycosylated form. And indeed, if we treat with glycosylating enzymes, this monomer collapses to a smaller size that perfectly matches this mystery band size. So this is our hypothesis, just from reading cell biology textbook. Maybe the normal TMEM106B with a short 3' UTR gets translated at the ER through Sec61, interacting with SRP, then goes through the secondary pathway, gets glycosylated in the ER, but perhaps the much longer 3' UTR somehow occludes this interaction and might get translated in the cytoplasm where it's unable to get glycosylated. It's just a hypothesis, but we're very interested in figuring out ways of changing the metabolism of the dimer. This just says so the take home here so far is that TDP-43 is doing more than just regulating cryptic splicing. it's this alternative polydilation. There's some interesting things. I'm very interested in TMAM106B right now. And that's it. I mentioned collaborators along the way. Thanks so much for inviting me. Happy to take questions.
Is there any studies? Are there any studies? CSF, introduction of CSF medication that abort or reverse ALS, for instance, cortisone or other therapy. CSF injection.
Inject CSF. So for the rare cases that have SOD1 mutations, if you inject into the CSF an oligonucleotide that downregulates your mutant gene, that's now approved for FDA. I don't know of small molecule therapies delivered to CSF.
Yeah, I mean, for instance, any nutritional ingredients that can help with neurologic disorders, nutritional, such as minerals or anything like that.
Yeah, I don't know of those studies.
Fantastic talk. I have two related questions. One, it seems like a lot of the disease pathology then is explained by loss of nuclear TDP-43, right? So what do you think is the cause for the toxicity in yeast then? And the related question is, does polyQ expansion PBP also cause toxicity in yeast?
Got it. Yeah, so we overexpress TDP-43 and it kills yeast cells. Is that relevant at all? I think what we're modeling is it's interfering with other RNA binding proteins. We're not modeling the disease, for sure, we're not modeling the disease in yeast, nor in those mice, actually. And we could really talk, I mean, I'm still trying to figure out why the ASO result in human. It turns out the PBP in yeast doesn't have a polyQ, nor does the mouse one, by the way. So polyQ, I think the polyQ thing, and some of our studies suggest that it's not the polyQ per se, but it's what the polyQ does to the context, so that it attacks into as an RNA binding protein. I think that's important. But yeah, I think we're like how we got to PBP1 is just to show like how it can antagonize its toxic effects. I don't know how to say if the cytoplasmic role of TDP-43 is more important or not. I think right now the loss of function seems like it should be really important, but it's out there in the cytoplasm. It must be doing. It's not inert.
Yeah, thank you.
Hi, have you looked at how in your ASO of Ataxin-2 and the mice, how that's affected accumulation of PINK1 and Parkin, or how that's affected mitophagy?
No. Do you think it would affect PINK1?
Yeah, I mean, if you're rescuing it, you might be increasing mitophagy. Like, have you looked at protein aggregation in the mitochondria in this model?
Oh, we looked at the spinal. No. We looked at the spinal cord for TDP-43, but yeah, we haven't looked for general aggregation, things like that. Yeah, that's a good point to think about.
Thank you.
Hi.
Hi.
So I also have two related questions. One is, it seems like what TDP-43 is doing is that it's regulating some sort of RNA metabolism. And when it's not there, you see all these polyadenylation defects, et cetera, et cetera. So instead of knocking it down, my question is like, have you tried forcing it out of the nucleus maybe, and then seeing whether it's the same effect as a knockdown? And in that case, what would be the exact mechanism of like, ataxia rescuing TDP-43? Is it pushing it back into the nucleus? And how is it helping in RNA metabolism?
Yeah, those are the great questions. Those are really great questions. We haven't done it, but you can mutate the nuclear localization signal of TDP-43 in an inducible way to force it to accumulate in the cytoplasm, and then it somehow sequesters the nuclear stuff. So there are other ways of doing that. That has the same, that also induces cryptic splicing events. You know, we've tried one experiment to knock down ataxin-2. We can't rescue the cryptic splicing events. It's possible that ataxin-2 is not going to rescue, or it's almost certain that ataxin-2 knockdown is not going to rescue all aspects of TDP-43 dysfunction.
And then have you looked at other cells instead of neurons?
So we're starting to look at muscle, just very, yeah. So I was mentioning these cryptic exons I'm interested in. Like we just started looking at muscle, and there's really exciting cryptic exon inclusion in. There's TDP-43 pathology in muscle, and there's a disease called inclusion body myositis. It's a TDP-43 pathology in muscle, and we see interesting things there.
Thank you.
Hi, really great talk. I was wondering, when the cryptic exons are expressed, why do you expect to find peptides rather than just longer unnatural forms of the protein?
Yeah, maybe I oversimplified. We would expect to see like a longer protein. Yeah, like a longer protein. We would see like if that. We're doing a lot of AlphaFold 2 modeling to see like those new sequences, if they stick out, if they're immunogenic, if we can make antibodies to them and stuff like that. So I shouldn't have just said peptides.
Got it. And
but there is in my field, like when there's repeats expanded, there can be this non-canonical translation, like even in different reading frames. So it's possible.
Right, the RAND translation.
Yeah.
But in this case, it's just a cryptic exon inside the protein.
Yeah.
So my follow-up question is, have you started making antibodies like the ones that Philip Wong made?
Yeah, so he made one against HDGFL2, and that seems to be a really good marker of TDP-43 dysfunction. I mentioned muscle. I think we found one that is expressed highly in muscle, could be interesting. Yes, we're starting to make antibodies to it.
One last question.
Oh, hey, Aaron.
Oh, hi.
You know, I've been thinking a lot about late recently because we've had some patients who were actually screening for lecanemab.
Can you tell them what it is?
A limbic, age predominant TDP-43 apathy. And it comes later in life, and it sits in the hippocampus, and it causes like an amnestic pathology, but it doesn't have the rapidity that we think about with ALS. And it looked like, I was just looking as you were presenting, like Nick Siegfried and his group are demonstrating that there is maybe some cryptic exon splicing in those cases as well. I think it's still emerging. But it's interesting to think about that contrast, because this is a very different part of the brain, you know, different type of, well, not that different than FTD perhaps, but it has a different flavor to the dynamic and the et cetera. So, you know, we were looking at things downstream of TDP, and you presented initially selective vulnerability. So the broader question baked into all of this is, you know, how are you starting to put all of this together? The fact that you have these different modifiers, yet we see this toxicity maybe shared downstream, and how is it that this fits within the context of different diseases with TDP?
Yeah, those are hard questions. One idea is that there are profiles, cryptic exon profiles. So even if we just look at, as you know, FTD, there's subtypes. So maybe there are profiles of these cryptic exons that are regulated in a differential way by TDP-43. We know that some of them are very sensitive to TDP-43 levels, like stathmin-2. You lower TDP-43 a little bit, stathmin-2 crashes down. UNC13A, it maintains itself until you really lower TDP-43, then it goes down. So maybe, like, depending on the extent of TDP-43 pathology, the type of pathology, you might get different profiles, and that might fit in. That's one idea. Yeah. It's also, yeah. Okay.